now?
Yes, please.
Okay, so first of all, thank you for inviting me the first memorial year-end meeting today. Actually, it's past 1 a.m. here in Japan, but I'm waking up from sleepiness by attractive thoughts so far. Okay, so today, I will talk about the importance of protein phosphorylation on metabolic regulation of hypoxic wax ester production in Euglena. So, as you know, Euglena accumulates a large amount of pamidon, beta-1,3-glucan as a storage substance under normal hypoxic conditions. And when aerobically grown cells just transfer into low oxygen conditions, you can then accumulate the wax esters, which can be promising alternative biofuels. The major component of wax ester is myristate C28. The hypoxic production of wax ester from pamidon is called wax ester fermentation that was found by Professor Inui in 1980s. It was quite unique pathway only found in Euglena, and especially as Masami said, the fatty acid synthesis is reversible beta oxidation, and the key enzyme of the pathway is a unique pyruvate NADP oxide reductase PNO that plays hypoxic production of acetyl-CoA instead of ordinary pyruvate dehydrogenase complexes. So the aim of our study is to identify the regulation mechanism of wax ester production in response to hypoxic conditions. So I'll show you some of our results briefly. So first, we carried out the transcriptome and the proteome analysis. And as shown here, so the changes in both transcription and translation levels of genes involved in wax ester metabolism were not so significant except under hypoxic conditions, except the PNO protein levels. So this result suggests the possibility of post-translational modification, including phosphorylation. Actually, the treatment of protein kinase inhibitor K252 shows a significant suppression of wax ester accumulation under low oxygen conditions, supporting the importance of phosphorylation on hypoxic wax ester production. So we next did the phosphoproteome analysis. And as a result, we obtained a total of 500 differentially expressed phosphoproteins in response to anaerobic conditions. And we found that 26 protein kinases are involved in the list. So we next focused on these protein kinases. By screening using the gene silencing of genes for these individual protein kinases, number 16 shows a lower accumulation of wax esters compared with mock control here. So we call this as wax ester synthesis regulation kinase, WSRK, and proceed further detailed analysis. So we measure the content of pamidon and C28 and fatty acids in the WSRK knockdown cells before and after hypoxic treatment. And as a result, the content of pamidon shows no significant difference between mock control and knockdown cell lines like this. On the other hand, fatty acid contents were significantly suppressed in knockdown cells as the green bars indicated. So therefore, WSRK does not affect the pamidon metabolism, and its functional point is accepted to be between glycolysis and fatty acid synthesis. In addition, metabolome analysis shows that the content of some amino acids derived from glycolysis were increased in WSRK knockdown cell lines as highlighted in red background. In contrast, the acetyl-CoA level in knockdown cells were markedly lower than that in mock control. So taken together, so conversion from pyruvate to acetyl-CoA was suggested to be the causal point of WSRK knockdown. So supporting this view, so previous phosphoprotein analysis shows that both PNO and PDH alpha subunit were phosphorylated, and their phosphorylation levels are varied in response to low oxygen conditions, shown here. So to confirm the phenomenon, so we then evaluated their phosphorylation levels by using their specific antibodies. As a result, WSRK knockdown strongly affects the phosphorylation of PNO, not PDH, under hypoxic conditions like this. So this result strongly suggested that the PNO is a downstream target of WSRK. However, so WSRK is suggested to be localized in the cytosol. So how WSRK finally phosphorylates mitochondrial localized PNO is an important matter to solve. So by the way, the previous screening of protein kinase brought us not only WSRK, but also more useful information. We found that number 25, showing high accumulation of wax ester, was an author of dual specificity tyrosine phosphorylation regulated kinase, DYRK families. So DYRK family has recently reported as a key regulatory protein kinases for triacylglycerol accumulation and starch degradation in chlamydomonas. Actually, so we found Euglena contains at least 3 DYRK family kinases, that is TAR1 and STD1 and STD2. Importantly, so knockdown of STD1 and 2 genes showed a significant increase in both palmiron and C28 content like this. Though the mechanism is now under investigation, our observation indicated that STD1 and 2 are negative regulators of palmiron and wax ester metabolism. So finally, so summarizing my talk today, I would say post-translational modification, including protein phosphorylation, is important to understand metabolic regulation, in some cases in Euglena. So here's the acknowledgement. Thank you for your attention.
Thank you. That was right on time, very interesting.